All right. Everybody, welcome to the second seminar of this semester of the Xylella Seminar. Thank you again for everybody for joining in. In the US, this week is Thanksgiving, so maybe difficult to get enough people from the US. Some people are traveling, but I appreciate that everybody showing up. So we have an exciting talk today. So Luis Moll is going to tell us about his research and his PhD. So Luis got a bachelor's degree from the University of Girona in Spain in biotechnology. In the same university, he got a master's degree in food biotechnology. And now he is into his PhD in technology that he's planning to defend in a month or a few weeks. So he has done an excellent work. You probably have seen his papers. He has been publishing a lot of his work already, so probably you can find it in publications. And his PhD he has like three advisors in his PhD: Ana Bonaterra, Esther Badosa, and Lydia Feliu. So he's keeping very busy working on these antimicrobial peptides. We also have the pleasure of having Luis visiting our university in maybe 2 or 3 years ago. I don't remember anymore. Maybe 2 years ago, and in the framework of the Euro sand cost action, and he did some work, but he's going to talk about it too. So we are looking forward to your talk. And again, as usual, after he finishes the talk, you can ask questions with yourself or write them in the chat. So Luis, we're looking forward to your talk.
Thank you very much for the presentation and for the opportunity for presenting this work for you today. So let's go straight into it. Before starting, I would like, as Professor Leonardo mentioned, most of the work has already been published. So all of the information that will be displayed can be checked in the papers that you can see on the screen. So feel free to screenshot it. And if you are curious about some details, you can also look into them later on. So we all know Xylella fastidiosa. It infects a wide variety of hosts, and it causes diseases in more than 100 of them, resulting in a very important economical impact. Initially, this plant pathogen was contained within the American continent, but since its first detection in Europe in 2013, several outbreaks have been detected, raising the importance of doing research on this pathogen. As we know, it is a gram-negative bacteria. It is rod-shaped, xylem limited. It has fastidious growth. It is transmitted by vectors. It forms biofilm, which correlates with the appearance of symptoms. And although it lacks flagella, it is able to move due to the presence of type 4 pili through a type of movement known as twitching. Generally, Xylella is classified in 3 major subspecies, which are Fastidiosa, multiplex, and pauca, although other subspecies have been proposed in the literature. They have also been classified based on the sequence type using MLST and 7 housekeeping genes. So the life cycle of Xylella fastidiosa, as mentioned, it is based on being transmitted by a vector. So once a vector carrying the pathogen feeds upon a xylem of a healthy plant, it can inoculate the bacteria. And in the first stage, the pathogen evades the plant defense system due to the presence of long lipopolysaccharides that are found in the plasmatic membrane. Then they start to multiply, attach to the surface of the xylem vessels, and form biofilm, which eventually can occlude them, resulting in the disease symptoms that we observe. Eventually, some of these cells or small clusters can detach from this biofilm and become planktonic, and eventually systematically colonize the whole plant. Eventually, a vector can feed upon the infected plant, acquire the pathogen, and transmit it to a new healthy host. In the EU, there are many regulations and directives that are in place, which follow an integrated disease management approach. Sadly, at the moment, there has not been a product that can cure or can completely cure infected plants, but there are many that are under research. And in this presentation, we'll be focusing on peptides. So peptides, what are they? They are sequences of generally less than 50 amino acid, cationic and amphipathic. They may present different mechanism of actions. And the one that has been the most studied is the one that you can see on the video on the screen, which basically is the interaction with the plasmatic membrane, resulting in pore formation or the disruption of the membrane, causing the death of the pathogen. Nevertheless, peptides can also interact with other targets, such as specific membrane proteins or lipopolysaccharides. Some of them, they can even translocate inside of the cytoplasm and, as you can see, interfere in many essential processes. So basically, this results with peptides having several activities, such as antimicrobial activity, antibiofilm activity, or interfere in processes such as motility. Peptides can also be applied to induce the plant defense system. So generally, in a normal plant, a healthy plant, what occurs is that when a pathogen is recognized, it is recognized due to the presence of specific sequences that are called PAMPs, or pathogen-associated molecular patterns, that are recognized by receptors found in the plasmatic membrane, which activate the pathogen or pattern-triggered immunity, resulting in a cascade and ends up with a defense response. Some pathogens have developed effectors to hamper this process, and plants have also evolved to counteract it and activate another system known as effector-triggered immunity. In both cases, it is activated the defense response at the localized level, and it can induce what is known as a systemic acquired resistance, resulting in the activation of this defense response through the whole plant. So basically, we can apply peptides externally to induce this system and make the plant to enter in a state known as priming, which basically makes it more resilient to subsequent infections. So the research groups I've been doing my PhD in, which are the LIPS and the FITSA, for some time they have been developing peptides to treat diseases caused by many plant pathogens. One of the most successful ones is BP100, which several modifications have been performed in sequence in order to improve its activity. For example, by introducing the amino acids, we can increase the stability and reduce the hemolytic activity of peptides. We can also introduce an acid chain to increase the specificity and activity of peptides. Or we can, for example, conjugate it with other sequences in order to enhance the properties of this peptide. So what about Xylella? When it comes to Xylella fastidiosa, at the beginning of this PhD thesis, there were almost no peptides described to be active. We had these ones, such a few examples would be secropine, indolecidine, and meganine, and it has been determined their antibacterial activity. So the LIPS and TITSA groups, in a previous work, they tested some of their own sequences and they assessed that they were active, that they had bactericidal activity against Xylella. But overall, there were no studies regarding the effect of peptides on other processes, such as antibiofilm, such as if they had antibiofilm activity, or if they could interfere in other key processes. So the objective of my PhD thesis was to identify peptides that target key processes in Xylella fastidiosa and its host for the leaf scorch control in almond plants. In order to do that, we basically followed a typical drug discovery process adapted to plant protection products, and which has the following steps. And we focused ourselves in two different groups of peptides, ones that had activity or direct activity against Xylella fastidiosa, and second one that acted as plant defense elicitor in the host that we chose, which in this case was almond. So let's start with the first group. To start, we need to select, design, and synthesize the peptides. So we did a literature research, and we checked for sequences that had been described to be active against gram-negative bacteria. Here you have the list of the pathogens that we looked for, specifically that they had antibacterial activity or antibiofilm activity described. Whenever the information was available, we chose those that had the lowest toxicity as well. these peptides are the ones highlighted in gray. The ones in white correspond to small modifications that we did to the sequence, the parental one that you can see on the top, in order to study their activity. So then we have to obtain the peptides. There are several mechanisms or methods to obtain peptides, but we decided to go with chemical synthesis, specifically some technique or method called solid phase peptide synthesis. As the name suggests, it is based on using a solid support, which is a resin. Here you can see it. And then between the sequence that we will be synthesizing and resin, we add a molecule, which is the linker, that acts as a bridge. Then we use a chemical approach, which is the FMOC turbotyl, which basically consists of protecting the amino acids at the N terminus with FMOC, which is a temporal protecting group. And in case the amino acid had a functional group, it was protected with a permanent group, which was turbotyl, bulk, PPF, or any other equivalent. So how do we do it? Well, we have the resin, the linker, and which is protected with FMOC. We remove it using a treatment with piperidine and DMF. And then we coupled the first amino acid using DIC, oxima, and DMF, generating the peptidic bond. We proceed to repeat these steps sequentially until we have all of the sequence. At the end, we remove the FMOC at the N terminus, and we cleave the peptide from the resin. And during this process, we also remove the permanent protecting groups, obtaining the sequence that we want. At the end, we analyze it by HPLC, and we characterize it using mass spectrometry to confirm that we obtained the peptide that we wanted. So after obtaining the peptides, we would move to proceed to the screening phase. We tested several activities. Here you have two of them. The first two, which are bactericidal activity and antibiofilm activity. For bactericidal activity, we did, it was a contact test coupled with a viability qPCR. So we put the peptide in contact with Xylella, and after 3 hours, we added PMA. PMA is a DNA binding dye that is not able to penetrate the membrane of the cells. So basically, it only interacts with the DNA of dead cells. After a photoactivation and DNA extraction, we proceed to do a qPCR, which only amplifies, or the only DNA that is amplified is the one from the cells that are alive. So we only are counting the cells that are viable. When it comes to antibiofilm activity, basically we co-incubated the peptide with Xylella for 5 days, and then we proceeded to the reading of total and tontonic cells. We did a staining protocol with crystal violet to read the amount of biofilm that has been formed. This looks quite easy on paper, but we had to set up a lot of conditions to do the screening to standardize it. So we selected regions, which protocols, how we should do the protocol, concentrations of peptides and cells. But the most important parameter or factor was selecting a model strain for the screening, because it would not be feasible to test a high amount of peptides with a lot of strains. It would take too much effort. So we selected 6 strains that were interest for us. And they were from different subspecies, Fastidiosa pauca multiplex, different places of origin, as well as different STs or sequence types. So for bactericidal activity, for each of these strains, we set up the calibration curves. We determine the limit of quantification. We also assess the susceptibility to antimicrobial peptides, some that we tested before. For antibiofilm activity, we measure the growth curves, the biofilm formation, and biofilm formation kinetics, as well as the susceptibility to antibiofilm compounds. In this case, it was N-acetyl L-cysteine. I don't have time to show you the results in this case, but basically the strain that we selected was Xylella fastidiosa, subspecies fastidiosa, EVF 5387, which was ST1, because it showed moderate susceptibility, moderate growth, and moderate biofilm formation. Then we also, for all of the peptides, we assessed the toxicity they had in eukaryotic cells. For plants, we used tobacco as a model. And basically, we infiltrated the peptide into the leaf. Sorry. And after 2 days, we measured the diameter of the lesion. We also assessed the hemolytic activity. So we obtained the erythrocytes from horse blood. We put them in contact with the peptide, and after 1 hour, we measured the amount of hemoglobin that was released, determining the percentage of hemolysis. So here you have the results for all of the activities that were tested for all of the peptides. As you can see, this is quite difficult to pinpoint which are the best candidates to keep on the track of this drug discovery process. So we did the principal component analysis. This allowed us to cluster the peptides based on their activity, being the most interesting one that you can see in green here, which is 1036, RHAK2, and Maglinin2, that showed to be or to present high bactericidal activity, high antibiofilm activity, and moderate toxicity. So then we would move to the next step, which would be lead optimization. In this case, we focus mostly our efforts on 1036, in which we did some amino acid substitutions that you can see here, that are the ones in white. We also selected some other peptides that we wanted to try, and we tested them together. And also we did with these peptides some conjugation by combining the sequences. We tested for these peptides for all of the activities previously mentioned, but we also now were interested in assessing the effect on the motility. So that meant that we had to assess the screening method for that as well. For the first step, which was assessing the previous activities, here you have the results. And I would only like to highlight this peptide, which is KSLWFD7, which showed high bactericidal activity at low concentrations, such as 3.1 micromolar. And regarding the modifications performed to 1036, it did not greatly improve or the activity overall, so we decided to stick with 1036 for the following experiments. So now we need to assess for all of the peptides the effect on motility. To do that, I went to the United States in our university in Leonardo de la Fuente's lab. And basically, we used a protocol that has been previously reported in the literature that consists on spotting the strains onto agar, and after a set amount of time of incubation, in this case 4 days, we observe if there's a fringe, which is indicative of twitching motility. So from these 6 strains that we previously assessed, we determined their motility through time. And interestingly enough, only Temecula L showed this typical fringe. The other isolates did not. And this fringe increased over time, as you can see in this graph. This was very interesting because these isolates, which are from Spain, we had previously tested them in almond. We inoculated it in this area, and we were able to detect them at distal points from the inoculation point, but especially below, which would mean that they have to travel against the xylem subflow. So they had to have some mechanism of movement. Based on that, in our university as well, we did test them under microfluidic chamber conditions, which basically try to imitate the hydrofluidic conditions of the xylem. We have some entry points and some outlets where we recollect the residues. In this case, we tested Temecula L, or we compared Temecula L with this isolate, the VF5387. What we observed was that both strains were able to move, but in fact, the VF5387 moved much slowly than Temecula L, which could explain the results that we observed on the agar plates. So based on this data, we decided to go with Temecula L as the model strain, and since microfluidic chambers, doing the screening using this technique would be technically difficult, we decided to do it on agar. The protocol was exactly the same, but the peptide was included inside of the medium. And then we spotted Temecula L on top of it. And after 4 days, we measured the fringe. We observed the fringe through 4 days. Here you have all of the peptides. We have a negative control with water BSA, which is a positive control at 6 grams per liter, it completely inhibits the motility of Xylella. So as you can observe in these images, the peptides did in fact affect the width of the fringe, as for example, you can see in this example. So we decided to calculate it or do a ratio between the fringe of the treatment compared to the non-treated control, obtaining this graph, as you can see here. This allowed us to pinpoint these two peptides, BP100 and FB7KSLW, which greatly affected the motility of Xylella, highlighting BP100 that reduced the motility by an 80%, 87%. Based on the previous results, we decided to confirm the activity on microfluidic chambers as well. So here we have the non-treated control and a negative control, which was FB7, that did not show activity during the screening process. As expected, to make the effect on Temecula, the motility of BP100, could be observed that it was reduced through time, confirming the activity of BP100 in this case. So at this point, we were curious about which was the mechanism of action of this peptide. And one of the initial hypotheses that we had was that the peptide was inhibiting the synthesis of the pili. So to test that, I went to the University of Córdoba and at the IES in Blanca Landa's lab, and we did some transmission electron microscopy experiments. So we took Temecula, we spotted it on agar and also on liquid media, and then we did some observations that basically the results obtained were the same for both. We were able to see that the type 4 pili was present in the non-treated cells as well as the treated cells, and that the frequency in which we found them, as well as the average length of this type 4 pili, was non-significant when compared to the non-treated control, which meant that the peptide did not affect the synthesis, but it would be affecting some kind of mechanism related to the function of the pili. Okay, so this would be, we will do a little stop here for the first group, and we'll be moving to the second group, which were the peptides with plant defense elicitor activity. So again, we need to select, design, and synthesize the peptides. For selecting them, we did a literature research, and we found these peptides that had been described to act as plant defense elicitors in other model plants, such as tobacco, tomato, and Arabidopsis. We also selected some other peptides that we found to be interesting, and we did some new conjugates. The synthesis of the peptides was we used the same technique or protocol as or method as explained before. But in order to determine the plant defense elicitor activity, we need to establish the conditions to do the screening. So here we go again, right? For the model, we decided to use flagellin as a model peptide, flagellin 22, which is a sequence of 22 amino acids of the flagellin, which is a subunit of the flagella. This sequence has been reported to be or to act as a plant defense elicitor in a lot of model plants. So since no information was available in almond, we had to select some markers. And how do we do that? Well, we did a treatment, we did an RNA-seq analysis, which allowed us to select some markers, which we will see in detail in a moment. Then we validated and optimized the screening platform and the conditions. And then we did the screening. So for the first step, we did a co-treatment of flagellin 22 using spray and endotherapy together. Then we sampled, we homogenized, we did an RNA extraction, and then we did RNA sequencing and analysis. This allowed us to identify several differently expressed genes, which then we classified in functions and pathways, which allowed us to see that most of the upregulated genes belong to defense pathways and were mostly focused on the salicylic acid pathway and abscisic acid pathway. Then we grabbed these genes and we transferred to a general scheme of the plant defense response, which allowed us to pinpoint some genes that could be of interest in, and we could use as markers. We tried to select some of each of the most representative pathways that we found. So salicylic acid pathway, abscisic acid, and jasmonic polyethylene. Of course, we did the design of the primers, we validated, and we validated the technique, showing that they worked. And then we decided to optimize the conditions of the screening platform. So if you remember, initially we did a co-treatment of spray and endotherapy, but we wanted to know how the gene expression changed based on how we applied the peptide. So we applied the peptide independently using spray, endotherapy, or we also infiltrated directly into the leaf. Basically, what we observed was that endotherapy was a strategy that resulted with the highest number of genes to be upregulated and the highest fold change. So was the application strategy that we followed to test in the following experiments. Then we were also interested in how the expression of these genes changed over time. So we sampled at different time points after applying, in this case, both cases was flagelling. And what we observed was that the genes were, the most number of genes that were overexpressed was at 6 hours. So we set up the conditions of the screening platform. We need to apply the peptides using endotherapy and sample at 6 hours. We applied all of the peptides that we selected and synthesized. Then we analyzed the data, and here you have the representation, which is basically a heat map of all of the marker genes and all of the peptides clustered based on similarity on the pattern of expression. And this allowed us to identify this group of peptides, as you can see in this blue square, that caused the upregulation of all of the genes, and the intensity was higher than the reference peptide that we were using, which was flagellin 22. So these could be good candidates to be further tested in the following experiments. So let's do a little bit of summary. We have the peptides with activity against Xylella. We synthesized them, we tested for these activities, highlighting 1036 as a very good peptide. Then we did a little bit of lead optimization and tested other peptides. And we also assessed the effect on motility in this case, which allowed us to identify KSLWFB7 as a peptide with high bactericidal activity and BP100 that inhibited the motility of Xylella. Regarding the peptides respond defense elicitor activity, in almond, we synthesized the peptides, we set up the screening platform, and then this allowed us to test several peptides, highlighting in this case FB7 due to its short sequence, which makes it easier to synthesize, and it caused an upregulation of the genes that were selected higher than flagellin. We also selected flagellin as a reference in this case. So now we could move to the in-house testing, right? Again, to do that, we need to determine how we want to apply the peptides, which strategy should we use, and then test them. So there are several methods that were available, and basically we were discussing whether to apply the peptide with the spray or endotherapy. We decided to go with endotherapy because the Xylella lives in the xylem, so we could more precisely apply the peptide and also apply the specific dose that we want, ensuring that gets inside of the plant. And then we had to choose which application strategy that we want to test. This was performed in parallel as we were doing the screening of all of the peptides. Okay, so we used BP178 as a model because in a previous work from the group, it was identified as being lytic against Xylella. So it was used as a model for the following experiment. So we have almond, which is the host that we will be using. In white, you have the points of inoculation of Xylella, and in the black squares, you have the points where we apply the peptide. The concentration of Xylella, we already set it up in a previous study. The application of the peptide, the concentration, we had to do a little bit of calculations based on in vitro data. So we know that the peptide to be active, it was needed this amount of concentration. So based on the amount of cells that we inoculate into the plant, we calculated the required amount that we would need. And after doing some calculus, this was what we ended up with. Regarding the application of strategies, we tested preventive, curative, and a combination of both. The preventive application consisted of applying the peptide 1 day before inoculation. The curative was 3 days and 7 days post-inoculation. And of course, the combination was doing both at the same time. The peptide, as mentioned, was applied in endotherapy, as you can see in the video. And basically, through 180 days, we sampled and monitored the plants, evaluating the population levels and disease severity. For population levels, basically, we analyzed 3 areas of the plant that you can see here, 8 centimeter segments, and we extracted the sap, and we added PMA, which was exactly the same protocol, in order to quantify only viable cells. So as you can see in the graphs, from the different treatment, application strategies that we used to apply BP178, the most efficient one was the combination of preventive and curative that caused the highest reduction in population levels, between 3 and 4 logs in some areas. Regarding the symptoms, we used this disease severity scale, which was already published. And as you can see, the best strategy, one of the best, was the combination again of both preventive and curative, with a significant reduction compared to the non-treated control. Interestingly enough, the preventive application of BP178 showed also a significant reduction. I don't have the time to show you the results, but in a later study, we deduced that, we find out that basically it was due to the activation of the plant defense response due to the application of the peptide. So now we will have the peptides that we selected during the screening separated in 2 blocks, or 2 sets of experiments. The first one consists of BP178, which is added as a reference in this experiment. KSLWFB7, which has high bactericidal activity, and BP100 that inhibits the motility. We have the concentration at which we have to apply them, and of course, the application strategy that we saw that was the most effective, the combination of both. So during 90 days, we assess the population levels, the disease severity, the leaf physiological parameters using this device, which is the Dualex. It measures leaf reflectance, and we also determine the occlusion of xylem vessels. Regarding the population levels, as you can see, all of the treatments caused a significant reduction in compared to the non-treated control. And especially it was significant for those that had bactericidal activity. When it came to the disease severity, all of the treatments, again, caused a significant reduction compared to the non-treated control, being the best one, BP100. Not only that, but as I mentioned, we measured the leaf physiological parameters using the Dualex, which measures chlorophyll, flavanol, and anthocyanin content, which in the case of BP100 showed results similar to a not inoculated and non-treated control, showing the protective effect of the peptide. So here we have a summary of the results, again showing that BP100 was the best peptide of this set. And we were curious about how the treatment of BP100 affected if it affected the conductivity of water well, that will never be precise. If the peptide was able to protect the xylem vessels, so if it was more or less occluded. So we sectioned part of the plant, and we did some observations using the scanning electron microscopy. Here you have a not inoculated control, a not treated control, which was infected with or inoculated with Xylella, and the ones treated with BP100. As you can see, the ones treated with BP100 show a similar look to the not inoculated ones, with some small elements such as some tyloses or some biofilm formation. But in the case of the not treated control, these were severely affected. If we quantify it, specifically 34% of the xylem vessels were affected, while in the non-treated control, only an 84%. Well, an 84% was occluded with some kind of biofilm or tylose. So again, it shows the protective effect of this peptide. So then we will have the second set of peptides, which is composed of 1036 as antimicrobial, flagellum 22 as a reference for plant defense elicitor, and every 7 as a defense elicitor that has a stronger effect than flagellum. In this case, we determine population levels, disease severity, and leaf physiological parameters. Again, all of the peptides cause a significant reduction in the population levels compared to the non-treated control, being the antimicrobial one, the one that caused the highest reduction. For disease severity, interestingly enough, the best peptide was every 7 that showed a higher reduction in disease severity and results a little bit closer to the non-inoculated control compared to the other treatments. So this would be another good candidate to keep on working on. So we reach this point where we have some candidates. Here we have all of the summary of the results in the experiments that we performed in the greenhouse. And I would like to highlight, as you probably can imagine, BP100, because it was the best one in reducing disease severity and showed similar values to the not inoculated controls regarding chlorophyll, flavonol, and anthocyanin content. And every 7 would be also a very interesting one due to its mechanism of action. That was by activating the plant defense system. And since it does not have bactericidal activity, it could potentially be produced heterogenously using bacteria. So here we reach a little bit of the end of my PhD thesis. Of course, there's still a long way going. And there's a little bit of debate of what should we do now, right? So should we move already of testing on field, or maybe we want to optimize a little bit more the conditions, for example, analyzing the effect on several strains as we are doing the vaccine right now. How we will obtain the peptides, so how we can scale up the production. Should we maybe try some combination of peptides to see if the activity increases, the overall activity increases in synergy? So there's a lot of things that can be done. And yeah, we will be reaching the end of the presentation. So I would like to thank everyone that has participated in this project, whether it be directly or indirectly. Of course, all of the funding agencies and the organizers of the seminars for allowing me to do this presentation. And yeah, so as Leo mentioned, basically, I'm finishing my PhD and I'm starting to look for a postdoc. So if by any chance someone is looking for someone, please get in contact with me. I would really appreciate it. Or if you have any questions of anything that has been displayed, very specific ones, feel free to write me as well. And just to end, thank you very much for all of your attention. And if you have any comments or questions, I will be more than happy to answer them or discuss them with you. Thank you very much.
Thank you, Luis. Great presentation. You have a lot of work done and very interesting results. So now, if anybody has questions, please raise your hand or you can write it in the chat.